see the numbers of participants beginning to increase.
So how many people have been coming?
You know, we've had in the like 70 to 100 people live, and then probably an equal number of people accessing the recordings after the fact.
Great.
So it's been a nice audience.
Yeah.
Hello, everyone. It's just the hour now, so we'll wait another minute for people to join us, and we'll get started very soon. Okay, let's get started. I'm Eric Nestler. I'm today's host. It is a great pleasure to introduce our speaker today, Dr. Jill Becker from the University of Michigan. I want to also alert you that the next speaker is Dr. Elizabeth Heller in 2 weeks from today. Please join us then. I am now going to turn things over to Céline Nicolas from Bordeaux, France, who will introduce today's speaker. Céline, take it away.
Thank you, Eric. So hi, everyone. It's my great honor to introduce Jill Becker today for the worldwide seminar on neurobiology of addiction. Dr. Becker received her bachelor and master degrees from the University of Kansas, and then she got her PhD in neuroscience from the University of Illinois. She is currently a research professor at the University of Michigan Medical School. During her career, Dr. Becker made a major contribution in understanding sex differences in and sexual differentiation of the neural systems mediating drug abuse and motivation. Her early research laid the groundwork for the studies by demonstrating that there is a sex difference in dopamine release from the striatum. This sex difference is affected by neonatal and adolescent hormone manipulation. Our research has gone on to elucidate the mechanism mediating the rapid and direct effect of estradiol on striatum of female. Our lab investigated the effect of psychomotor stimulants on behavior in rat and how ovarian hormones act in the brain to influence drug-taking behaviors. Dr. Becker has been honored by many awards, including the Health Education Visionary Award. And today she's going to share with us her expertise in sex differences in drug addiction in animal model. Jill, whenever you are ready.
Thank you so much. Thank you for that nice introduction. Thank you to Eric and the rest of the crew for inviting me to do this. I'm really looking forward to telling you about my research. So I'm going to share my screen. And are you seeing the
Yes, that looks good, Jill. Yep, ready to go.
Okay. Okay. So yes, I'm going to tell you about sex differences in addiction as we've been studying rodent models. And I hope you'll stay with me here. So basically what I'm going to tell you is how sex differences in the brain occur and what that means for the reward system. And then I'll talk a little bit about estradiol modulation of dopamine release and stimulant drug motivated behaviors. And then some of the animal models for addiction that we've used trying to get at some of the mediating effects for sex differences in addiction. And then finally, I'll wrap up with some of the more recent work we're doing, trying to elucidate in chronic preparation fast scan cyclic voltammetry to look at sex differences in the neural mechanisms in rats. So let me begin by telling you that the chromosomal sex determines what gonads animals are going to have, and the hormones produced then influence sexual differentiation of the brain and their behavior as well. But there are also effects of the XY chromosome. The presence of the SRY gene within the brain has specific effects on sexual differentiation, and there are effects of hormones and proteins produced by the placenta that affect sexual differentiation. But in general, if you're female, you don't have an SRY gene and you get ovaries, and hormones are not being produced in early stages of development. If you're male, you have this gene called SRY, the testes determining factor, that gives you testes, and testosterone produced by the neonatal testes acts on the brain to result in sexual differentiation of the hypothalamus and areas important for sexual behavior. So this is the early period I just told you about, with the peak in testosterone during the perinatal period. And then again, at puberty, there is an increase in hormones that activate specific behaviors in males and females. What we know about sexual differentiation of the brain for motivation and the influence of estradiol in the dorsal striatum is it's the absence of hormones in females and the presence of hormones during the pubertal period that are necessary for the sensitivity to estradiol that I'm going to tell you about. Before I start talking about sex differences, I want to make sure we're all on the same page, because not all sex differences are bimodally distributed, which is to say, you don't always have male typical sexual behavior, for example, and female typical sexual behavior, two behaviors so different you can't measure them on the same scale. Those are what I call qualitative differences. There are also quantitative differences, which is to say males and females exhibit the same behavior, and you can measure it on the same scale, but the means are significantly different. And things like spatial function in humans and verbal behaviors, where females tend to be better at verbal behaviors, males tend to be better at spatial behaviors, but you can measure them on the same scale, and it's a mean difference. There are also population differences, which is to say both males and females exhibit a behavior, but one sex exhibits the behavior in a larger number of individuals. These things, I think, are caused not necessarily by the processes of sexual differentiation that I talked about in the last slide, but more about things like stress during development or the availability of an enriched environment can shift population differences. Then we also have these latent sex differences, where you have males and females exhibiting the same function, but the underlying mechanism is different. And the classic example of this is Debbie Van Gasser and Rita Valentino's discovery that the CRF receptor uses different intracellular signaling processes in order to mediate the same response to CRF. You can think about these. Adrian Beltz and Annalise Beery wrote this article with me in Neuropsychopharmacology that also talks about how you can think about these different types of sex differences in a more quantitative way and use it to design your experiments and analyze your data. That's a great paper if you're interested. So when we think about sex differences in the reward system, you can think about all different types of neurotransmitter functions, because the dorsal striatum that I'm going to be focusing on is only part of the story. So I always put this slide up to remind me to tell you that I know that there are many other neural systems involved. And even though I'm focusing on the area of the midbrain that projects to the dorsal lateral striatum and the nucleus accumbens in the talk, I'm also aware that there are sex differences in amygdala and the lateral hypothalamus and hippocampus and prefrontal cortex that are mediating many of the subtle effects that we're seeing on sex differences in reward. So I'm going to summarize a whole lot of research with this one slide and then give you a couple of examples. So, and this is something I've been studying since I did my dissertation many years ago with Domingo Ramirez. And what I discovered is that estradiol enhances dopamine release in the dorsal striatum of females, but not males. So amphetamine-induced striatal dopamine release varies with the estrus cycle in vivo and in vitro. Ovariectomy attenuates the response to amphetamine or potassium or cocaine, and estradiol treatment is sufficient to reinstate that response. And this, you don't see an effect of castration in males, and you don't see an effect of estradiol on these measures in males. I've also shown that estradiol acts directly on the striatum to enhance dopamine release, and I've shown this in vitro and in vivo. We believe, and have Paul Mermelstein's lab has shown that there are membrane receptors for estradiol. So we're going to call these MERs, or membrane estradiol receptors. Finally, I've shown that estradiol rapidly downregulates D2 dopamine receptors in the dorsal striatum of females. And I'll just give you a couple of examples of the direct effects. So Eric Song with Liz Peckham and Hongyan Yang showed that estradiol is acting directly in the striatum to enhance amphetamine-induced dopamine release using in vivo microdialysis. So this is a dialysis probe here. And so this is, if you ovariectomize a female rat and just give oil, you get this a small but significant, it's 1,000%, 1,500% increase in amphetamine-induced dopamine release. If you give estradiol subcutaneously or estradiol via the dialysis probe, you get an enhancement of the response relative to the ovariectomized control. So this shows that estradiol is acting directly in the striatum to mediate these effects. And then Terry Bezet showed quite a few years ago, when he was doing a postdoc with me, that estradiol rapidly downregulates D2 dopamine receptor binding in females. So this is the ovariectomized autoradiography of D2 dopamine receptor binding in females after ovariectomy, and this is 30 minutes after estradiol. There's a significant downregulation. So estradiol is acting in the brain by multiple estradiol receptors. There's ER alpha and ER beta that can be nuclear receptors as well as membrane receptors. And then GPER1, I'll actually be telling you a little bit about today because it also seems to be a significant player in the dorsal striatum. So all of these receptors can act rapidly on neural activity. And it's not just the brain where this happens. So that happens also in the heart and the bone and fibroblasts and in breast cells. So throughout the body, you're having these rapid effects of estradiol that are important for cellular function. So this is just a cartoon to show that estradiol can go into the cells to bind the classical nuclear receptors that induce protein synthesis. But there's also GPER1 on the membrane, and then ER alpha or ER beta coupled to metabotropic glutamate receptors to mediate rapid signaling effects. And Paul Mermelstein's group and Paul Masevich's group at UCLA, Dr. Mermelstein is at the University of Minnesota, have shown these rapid effects in a number of elegant ways. So the model that I'm working with as I look at the effects of estradiol on dopamine release and dopamine-mediated behaviors is that estradiol is acting on medium spiny neurons found in the dorsal striatum on membrane receptors. And it has rapid effects to downregulate D2 dopamine receptors, as I've just shown, and it results in a decrease in GABA release, attenuates GABA release. And it's this release of inhibition that mediates enhanced dopamine release. So the dopamine cell bodies are in the midbrain. They project to the dorsal striatum where GABAergic function is modulating their release of dopamine. And Kristen Schultz showed this working in my lab, where we did an overexpression of ER alpha in the dorsal striatum. And in the control gene, just the housekeeping gene, with estradiol is attenuating potassium-stimulated increase in GABA in dialysate. If you give the estradiol to animals that have an overexpression of ER alpha, you see an even greater decrease in the potassium-induced GABA release. And so you can see that here clearly, that estradiol attenuates GABA release, and overexpression of ER alpha enhances the attenuation. Kristen also showed that if you give the metabotropic mGluR5 antagonist MPEP, you can prevent the effect of estradiol to attenuate GABA release. So it's a decrease of the attenuation. So this is the saline response. And this is the response in the presence of MPEP. So we believe that shows that MPEP is the effects of estradiol are mediated by the metabotropic glutamate receptor 5. And we did a similar thing. Eric Song showed that if you can block the effects of estradiol to enhance amphetamine-induced dopamine release. So this is estradiol-induced dopamine release given systemically. If you give the ICI, which is an estradiol ER alpha, ER beta antagonist, you can prevent the enhancement by estradiol. And if you give MPEP systemically, you can block the effects of estradiol given intrastriatally. So MPEP is attenuating dopamine release as well as GABAergic effects mediated by estradiol, supporting our hypothesis that this is a membrane receptor effect mediated in part by the metabotropic glutamate receptors. Now, we've shown the response to amphetamine repeatedly, but all of our behavioral tests were with cocaine. So we thought it would be important to look at the effects of cocaine on the mediation by estradiol. And so we had ovariectomized female rats and castrated male rats. And we had dialysis probes in this dorsal striatum and in the contralateral nucleus accumbens. And what that allowed us to do is to give oil, the control, or estradiol to both male and female rats. And you can see in the dorsal striatum, as I've been showing you, estradiol enhances a cocaine-induced increase in dopamine in females, but there's no effect of estradiol in males. And the response in males is, the peak response is about the same as the estradiol-stimulated response. Surprisingly, in the nucleus accumbens, we didn't see an effect of estradiol in this test with a microdialysis probe that on examination turned out to be at the border between the nucleus accumbens core versus shell. We did see a sex difference in that there was lower cocaine-induced dopamine increases in nucleus accumbens compared to females compared to males. And estradiol did not enhance the effect. Well, not to be dissuaded by those important results, Katie Yost, when she was doing her dissertation in my lab, decided that it would be important to look specifically at the nucleus accumbens shell and determine whether you could see effects of estradiol on stimulated dopamine release. So she used fast scan cyclic voltammetry to, so the probe is in the nucleus accumbens shell. The stimulating electrode is in the ventral tegmental area, so stimulating the cell bodies. And animals were treated with estradiol. Then we stimulated the cell bodies, gave animals 10 milligrams per kilogram cocaine, and repeated the stimulation. So we can look at the peak dopamine release as well as the reuptake of dopamine. And I'm just going to tell you about the peak dopamine release. So what Katie showed is that if you give estradiol in females, you get an enhancement at the stimulation parameters that really drive the response. But in males, we did not see a significant effect of estradiol. We then went on to ask which receptor subtype is mediating this effect, and we were concentrating on ER alpha and ER beta. And so the ER alpha agonist is PPT, and the ER beta agonist is DPN, same parameters. So here's the effect of estradiol I just showed you. With the ER alpha agonist in the nucleus accumbens, we did not see an effect, but with the ER beta agonist, we did in females. In males, we did not see an effect of any of the agonists. And we saw a similar effect at both stimulation parameters. So that shows that estradiol can affect nucleus accumbens shell response, but it seems to be a localized effect in the nucleus accumbens shell. And you need to really drive the cells in order to see the response, indicating that it's something mediated by enhanced firing. So what I've told you so far is that estradiol is enhancing dopamine release in dorsal striatum and is acting directly on the dorsal lateral striatum in females. You see a rapid downregulation of D2 dopamine receptors. This is mediated by the medium spiny neurons, and mGluR5 appears to be important to mediate the effects of estradiol. And there are rapid effects in the nucleus accumbens shell on dopamine release, mediated by ER beta. So what does this mean for other types of motivated behaviors? And obviously, I'm going to tell you about sex differences in drug taking. So why would there be sex differences in motivation? I always like to step back and say, why would there be sex differences in whatever it is we're looking at? And I really think that the fundamental difference in maternal motivation is a big piece of why there are sex differences in drug taking behavior. And Jane Taylor and I wrote a chapter about it quite a few years ago now. But it's the rapid formation of the mother-infant bond, which is estradiol dependent in rodents in particular, where it's been studied, would mediate a more rapid attachment to drugs of abuse, is the hypothesis we put forward. We also find sex differences in motivation for reproductive behaviors and in the modulation of energy intake, where estradiol decreases feeding. So the neural systems mediating these motivated behaviors are different in males and females. Okay, so sex differences in drug abuse and addiction is really more than just how many individuals. It's not just a population difference. More men tend to be addicted than males, but the difference between males and females is rapidly becoming different, not different. And if you look at the pattern of drug taking, what you see is that the time from first exposure to a drug to women presenting for treatment is shorter for females than for men. And this is true for all classes of drugs. Females present consuming more, even though the amount of time has been shorter. And the reasons for initiation of drug use are different in males and females. Women report self-medicating for anxiety and depression, while men and boys tend to take drugs to be part of the group, and to engage in risky behaviors with their friends. So you see, this is just a summary of the aspects of addictive behaviors from a review I wrote with George Koob a few years ago. And so just basically what I just told you, escalation is more rapid. There are also greater side effects and withdrawal effects in females. And I'm not going to go through all of this, but it's true for all classes of drugs of abuse. And we see the same patterns in rodents, which is to say, while there are no doubt cultural effects on sex differences in drug-taking behavior, the fact that you see the same things in rodents as in humans indicates, to me at least, that there are biological factors influencing these. So what do we see? What we see is that if you let animals self-administer a drug like cocaine, so that this animal is going to nose poke and then get 0.3 milligrams per kilogram per infusion cocaine. And we had 4 groups in this experiment, ovariectomized females. Half of the one group received estradiol 30 minutes before the test, the other got oil. Then we have intact males who are sham operated and castrated males. And this was just to see, is there effect of testicular hormones? And we don't see an effect of testicular hormones on self-administration of cocaine, but we do see that estradiol treatment prior to self-administration enhances acquisition and enhances drug taking in females, but not males. Here's the data that show it doesn't work in males. Lisa Jackson, when she was working with me for her dissertation, showed that if you give castrated male animals estradiol, these are the estradiol treated females again, showing that you get enhanced acquisition and drug taking after estradiol in ovariectomized rats. But the castrated males are actually right in the middle. And when they give them estradiol, it doesn't affect their acquisition or drug taking behavior. So again, estradiol is not affecting this in males. If you look at breaking point in females, so each dose of a drug an animal receives results in an increase in the response required for the subsequent dose. And so at this point, 50 nose pokes, the animal says, that's enough, I'm not going to work anymore for cocaine. I don't want it at 0.3 mg per kg, even though I'm getting estradiol. So at this low dose, estradiol doesn't affect the cocaine taking behavior. But if the dose is increased to 0.4 milligrams per kilogram, animals who receive estradiol have greater motivation. So they're working harder, making up to 160 nose pokes in order to receive cocaine before they quit. And yeah, it's also true at a higher dose. So estradiol enhances the motivation to take cocaine in ovariectomized females. So the next thing we wanted to try to do is figure out which estradiol receptors are affecting drug-taking behavior. We saw effects of ER alpha and ER beta on dopamine release in dorsal striatum and nucleus accumbens, respectively. But do we see the same effect in preference for cocaine? So we started by using a conditioned place preference apparatus, pairing one side with cocaine and one side with saline. And we put the estradiol receptor antagonist 182780 into the dorsal striatum. And when we did that looking at amphetamine-induced dopamine release, it blocked the effects of estradiol. So we thought that would be a good bet. And to our surprise, if we look at females here on the right, you see that this is pre-pairing. And then after pairing with cocaine, the cholesterol-treated group preferred the side of the chamber paired with cocaine. The animals treated with ICI 182780 also preferred the chamber paired with cocaine. So we did not see an effect of the estradiol receptor antagonist in females. In males, this is the controls. They showed a preference for cocaine if they had cholesterol treatment in dorsal striatum. But to our surprise, ICI blocked the preference for cocaine in males. So this was surprising to us. And we then, this was Jackie Quigley's dissertation, and she went into the literature and discovered that ICI, not only is it an ER alpha, ER beta antagonist, it's also an agonist at GPER1. So she said, I'm going to see what happens to GPER1, see if it does the same thing. And it did. So the females, again, no effect of GPER1 on preference for cocaine. The females preferred the cocaine paired chamber. Again, the cholesterol males showed a preference, but the animals who had G1 in their dorsolateral striatum did not prefer the cocaine paired chamber. So for the first time, we've seen an effect of an estradiol receptor agonist in males, and it's the opposite of what we would have expected. Jackie also gave G15, the GPER1 antagonist, and it enhanced the preference for cocaine in males for a dose that was not previously preferred. These are data reported in her Hormones and Behavior article. So what we see is that cocaine preference was decreased with GPER1 activation and increased with GPER1 inhibition. So that was a pretty surprising result. And we'll come back to see what GPER1 is doing more in a few minutes. So one of the things we've been trying to do as we look at self-administration paradigms is develop paradigms that tend to be or could be interpreted as giving in animals a more normal-like environment. It's not really normal, but at least it's not, the cocaine's the only thing you can do, because quite frankly, if you were put into a chamber and you could do, only thing you could do was get cocaine, you'd probably give yourself cocaine, because why not? So we thought maybe rats are like that, and we should give them a choice. And so in this experiment, you nose poke in one hole and you get cocaine, and the other one you get these tasty banana flavored pellets. And early during the sessions, when animals are just learning about this choice paradigm, everybody prefers the pellet. So the number of pellets earned is significantly more than the number of cocaine infusions received. The open circles are the animals who continue to prefer pellets throughout the experiment. The dark circles are the ones that come to prefer cocaine. So then you can see at week 7, some animals who prefer pellets only work for pellets. Animals who prefer cocaine, well, except for this one male, only work for cocaine. So a clear preference has been developed. And if you look at the rate at which the preference is developed and the number of animals that prefer cocaine over pellets, what you see is that more females prefer cocaine than males, and they acquire this preference more rapidly. So we've replicated this finding in another study. And in the second study, we also looked at dopamine release during week 2 versus week 7 in the nucleus accumbens. And again, this is microdialysis, so it's the core shell border. So some animals actually never really take a lot of cocaine. That's sort of surprising, considering the idea that cocaine's better than peanut butter, but it's true. So in these animals, you can see early on, they have a significant increase in dopamine release induced by cocaine, and it's the same during week 7, and they haven't received much cocaine. The pellet-preferring animals also show the same increase in dopamine release in the nucleus accumbens early and late. And this is true even though many of these animals received as much cocaine during the sessions where they're not having to make a choice as the cocaine-preferring animals do. So it's not just that these animals got didn't get as much cocaine. The cocaine preferring animals, you can see that this is the early response, and that by week 7, there's a significant attenuation of cocaine-induced dopamine in the nucleus accumbens. And we had an equal number, basically, of females and males in this cocaine preferring group that we did microdialysis on. And so what you can see is that we saw the same pattern of a decrease in cocaine-induced dopamine in females and males, suggesting that the difference between males and females may not be a mechanistic one, at least at this level of analysis, but a population difference with more females developing the preference for cocaine. So if we go back now and look at GPER1 and cocaine self-administration, animals, again, they're nose poking for cocaine. Then after a week on FR1, we put them on a progressive ratio schedule for 2 weeks. Then they received G1 or cholesterol into the dorsal striatum and continued on a progressive ratio schedule for another 2 weeks. Then they underwent extinction, and then we looked at reinstatement. And this is published in Biology of Sex Differences, part of Dr. Quigley's dissertation. And so what we showed is that, again, we see that females have greater motivation for cocaine than males do. So there's a sex difference prior to manipulation. Then half of the females received G1 in the dorsal striatum, half received cholesterol, and the same for the males. And what we saw is that the G1 treatment actually enhanced the motivation for cocaine in females to a greater extent than the cholesterol treatment. And they're, they're still responding at about 100 responses for breaking point. Well, the males have a lower breaking point, and G1 did not enhance it. So this just summarizes the difference here. So unlike what we saw with G1 activation of GPER1 with conditioned place preference, for cocaine motivation, we're seeing that it enhances it in females, but not males. So the bidirectional effect of GPER1, males versus females, and the liking of a conditioned place versus wanting the drug cocaine. Jackie also showed that if you look at reinstatement after extinction, G1 enhances reinstatement induced by 10 milligrams per kilogram cocaine. And these groups were balanced so that half of them had received G1 previously and half had received cholesterol. So it's not an effect of prior treatment. So G1 is affecting motivation for cocaine and reinstatement of cocaine in females, but not males. Trying to look at, again, more things that might be happening to humans. We've looked at early life stress using restraint of females during the last week of gestation, and then testing drug-taking behavior in adult animals. And what we see is that prenatal stress enhances acquisition and cocaine intake in male animals, but not females. And the same treatment, if you look at motivation for cocaine, prenatal stress increases the proportion of females exhibiting the greatest motivation. So this just shows that the proportion of the population, again, of females that are highly motivated to receive cocaine has been shifted. And one other paradigm to tell you about is the effects of social housing on self-administration behavior as a potential model of social support. And what Crystal Westenbrook showed is that individually housed females take more methamphetamine than socially housed animals. And if you give them oxytocin, you see that for both the individually housed and the socially housed animals, there's a significant decrease in motivation for cocaine. So that social housing is potentially important for decreasing motivation in females. And you can see that here by just looking at the proportion of individuals with really high breaking points. The social housing didn't really decrease the animals with extremely high breaking points, but it increased the proportion of animals that had very low motivation for methamphetamine. And the effects of oxytocin were to, this is during the response prior to treatment with oxytocin. If you then look at treatment with oxytocin, what you see is that the highly motivated animals after 2 weeks of treatment had a significant decrease in motivation for cocaine, as did the intermediately motivated animals. So oxytocin appears to influence motivation. Okay, so more recently, we've been trying to, we have been working with Cindy Chestik's lab using a new chronic electrode to measure dopamine with fast scan cyclic voltammetry. And so we're recording from the nucleus accumbens and what I'm going to show you, and stimulating in the ventral tegmental area. So we're using a 9 millimeter long electrode that has fibers at 120 microns from each other. And there are 16 fibers that are projected, lowered into the nucleus accumbens and left there. And Paris Patel is the brilliant person who makes these and helps us with a lot of the recordings. So what you can see here is that if you look at the stimulated response, and this is just an example from an animal of the recording. These are just the 16 different channels. Obviously, this one's not working. Dopamine is right here with the little green dot. And if you give it 60 hertz for 30 pulses, you get a small response. If you give 120 pulses, you get a much greater increase in dopamine. This is just a color plot. And thanks to Dan Jacklet for providing these images. We leave the electrode in place and then decalcify the skull so that the slices are then sectioned with the probe in place, and you can actually see the fibers. You can see these are 2 of the fibers out of 16, and this is just a Nissl stain from one of the earlier versions of the electrode that we published this in the Journal of Neural Engineering. So the kinds of data that we are seeing, Dan Jacklet has been working on the algorithm so that we can do this much more efficiently, because with 16 channels, as you might imagine, there's a lot of data. But what you can see is that we get the maximum increase in dopamine increase after the electrical stimulation. We can also measure the rate of dopamine increase, and we can measure the area under the curve as the total increase in dopamine. If we look at the effects of social housing, this is just an example that Yvette Gonzalez of the data Yvette Gonzalez has been collecting in the lab. What we've been doing is we're socially housing versus individually housing males and females, and looking at how that affects the VTA stimulation of dopamine release. And then animals are given 3 different doses of meth for a cumulative dose of 1.5. To look at potentially sensitization of dopamine release. So this is the basal response in the absence of meth. And then again, this is the increase from basal. We do see a floating increase in just basal recording with methamphetamine, but you also see an enhancement of the response to meth in some individuals. These are just examples and not intended to tell you what the groups are doing. And I should also mention that Brandon Luma and Noah Leonardo, as well as Stan and Yvette, have been working on how we analyze these data. Part of what we're trying to do right now is make it more efficient. But this just gives you an example of the response of 7 of the different fibers that are present in the nucleus accumbens. And you can see that while these are responding similarly, not all fibers do respond the same. And then Noah has been slicing in place. This is the green is tyrosine hydroxylase, and then we've got DAPI, so you can see the neurons. And these are the fibers in the nucleus accumbens. This is in the shell. We're staining with calbindin to determine shell versus core. And what you can see is that the fibers do very little damage, and we're in tyrosine hydroxylase positive brain regions here. So what I've been telling you about is that there are sex differences in addiction and in the mechanisms mediating addiction. So we see that females respond more dramatically to cocaine, and this is in part mediated by estradiol. We see population differences driven by things like prenatal stress or the just preference for cocaine versus pellets. And we're trying to determine the extent to which there are latent underlying mechanism differences as well. So the initial response to cocaine and amphetamine is greater in females. Effective estradiol is qualitative difference, and the population differences in the vulnerability to addiction. So what I've told you is that estradiol enhances stimulated dopamine release and the acquisition and escalation of drug taking. Estradiol acting at GPR1 is attenuating the preference for cocaine in males, but enhancing motivation for cocaine in females, telling you that self-administration of drugs versus conditioned place preference are clearly working on different neural systems and telling you something different about how these drugs and how estradiol is acting. Stress enhances drug taking, but males and females are differentially affected by prenatal stress. Social housing can attenuate motivation for meth in females. And this is probably important for thinking about treatment of addiction in both men and women and the role of social support. And oxytocin can ameliorate the motivation for methamphetamine in females. So I hope you've seen that by looking at females, you may discover important insights that are relevant for addiction in both males and females, because these are fundamental differences in how the brain is organized. And understanding sex differences is going to help us understand addiction and the neural mediation of addiction in both sexes. So I've tried to mention the individuals who were responsible for the work I told you about as I went through it. Thanks to NSF and NIDA for funding. If you're interested in sex differences, there's a great group called the Organization for the Study of Sex Differences. Cynthia Chestic, Dawin Sai has been helping us with the histology. Huda Kil, Terry Robinson, of course, Paresh Patel's a great resource for us. So thank you, and I'm happy to take questions. This is the undergraduates working in the group, and then my core lab group of graduate students and technicians. So thank you.
Thanks so much, Jill. That was great. Thanks for such a great overview of this important field. If you could stop sharing, that way we could see one another.
Okay.
And we can start taking some questions. I'd like to encourage people to put questions in the Q&A function at the bottom of the Zoom screen. In the meantime, I have a question, but Celine, did you want to go first?
I can go first, up to you.
Yeah, go ahead. You go first.
Okay.
I'll ask my question next.
Thank you. Thank you so much, Jill, for the very interesting talk. I have 2 questions. One naive question. When you do ovarectomy and you give estradiol, which phase of the physiological estrous cycle do you mimic?
So giving estradiol, concentrations of estradiol that we see in the serum after the dose we use, which is 5 micrograms of estradiol benzoate, mimic the response that the increase in estradiol you see at estrus. And in all of our treatments, if we give 5 days of estradiol, that's obviously not very physiological, but we always give animals 2 days off before we start treatment again in order to allow the estradiol receptors to reset. And I think that's an important thing. If you chronically treat with estradiol, you actually see something different than if you just give it acutely like we do.
Okay, interesting. And my other question was, so you gave a very nice overview of the role of estradiol in the dopaminergic function in the striatum, but we also know that there are other ovarian hormones player in the system. So what is the role of progesterone in the effect that you showed?
So we've done a couple of things with progesterone. If you give estradiol and progesterone simultaneously, rather than priming for estradiol, you actually block the effects of estradiol to enhance drug-taking behavior in female rats. So that's one aspect of it. But if you give estradiol and then you're basically priming for progesterone receptors, so 3 days of estradiol and then progesterone, and you look at dopamine release, you see basically the same effect as we get with just estradiol alone. So it's not a simple answer. It's not just that progesterone always attenuates the effects of estradiol. If the animal is primed and there is an increase, and priming for estradiol means you actually increase the number of progesterone receptors, then you see a different effect.
Okay, thank you.
That's a good question. Thanks so much.
Eric, you can go ahead if you want.
Yeah, thanks, Celine. And Jill, I know that there's been an effort also to understand the chromosomal contributions to sex differences. And what is your sense of that and the status of the field, efforts to identify sex differences that are not due to hormonal mechanisms, but due to expression of other X or Y linked genes?
So I think, there's the work of Eric Villain suggests that the SRY gene is expressed in tyrosine hydroxylase positive neurons in the substantia nigra and ventral tegmental area. Mm-hmm. So, well, I'm not sure that it's relevant to the effects of estradiol I've just been talking about. It may be playing a role in the GPR1 effects we saw in males, and I haven't been able to get into that, but it's definitely modulating dopaminergic function in males.
No, it's so interesting. And then you addressed this to a certain extent. The discovery of GPR1 is obviously really exciting. So I remember when you and I were in training, there were always these rapid effects of gonadal steroids like estrogens, and they were really not well understood. Now we have a basis for it. What is your sense of the relative contributions, if you look at the effects of estradiol, like what aspects are mediated transcriptionally through ER receptors in the genome? To what extent are they mediated through GPR1, and to what extent are they mediated through the other action you talked about, which is the connection to the metabotropic glutamate receptors, I think you said? Mm-hmm. Yeah. Right. What is your sense of that, and how does your lab go about parsing out those mechanisms?
Well, that's what we're on the hunt for right now, is because we were just blown away when we found this GPER1 effect. It was not what we were looking for at all. And there are lots of people in the neuroendocrinology field who are not really happy to see this be an important contribution, but I think it is. I think it's, and part of what we need to figure out now is the extent to which the estradiol receptors are working together or in isolation. So what we know is GPER1 actually modulates the effects of ER alpha and ER beta in different areas of the brain, and in particular in hippocampus. And so that's the next thing we need to do, is parse out, are these effects solely mediated by ER alpha or ER beta or GPR1, or are the effects of estradiol being influenced by whether these receptors are present in the same or different cells? To what extent are the receptors collaborating or working antagonistically? So that's it's a really important question that we actually, I wish I had the answer for.
Sure. No, absolutely. Yeah. Celine, I could keep on going, but I don't want to hog the Jill's attention. Did you have other questions?
I don't have other question, and I don't see any question on the Q&A box. So if you have other question, I think you can go ahead, of course.
Okay, fantastic. Yes, so let me keep going. So, Jill, as you know, we were late getting to the question of sex differences in responses, for example, to drugs of abuse. And I've been struck by the dramatic nature of such differences. So just by doing the experiments and including female rodents in all studies, the biology is just incredibly compelling, basically supporting everything that you've been saying these years. I was wondering if you could say a bit more of something you alluded to, and that is, what do you think the evolutionary advantages are of these fundamental differences in reward circuitry based on responses to gonadal steroids in males versus females? It's so fascinating to think of the magnitude of differences that are seen.
Right. Well, I really, if you think about the expression of maternal behavior, what we know is that during pregnancy, estradiol increases and is necessary for the expression of maternal behavior. And that maternal behavior happens as soon as the female gives birth. She immediately forms this rapid mother-infant bond. And that mother-infant bond is dependent on dopamine release in the nucleus accumbens and oxytocin release. So those two things have been shown in the rat and in the ewe to be what are necessary for the formation of the mother-infant bond. I believe those factors are also important for the formation of the attachment to other things like drugs of abuse or high caloric foods if you're a food addict. So I think that the system is actually designed for females to form rapid attachments, and that drugs of abuse are sort of using that system. I also think that could be why oxytocin can ameliorate the effect, is that because oxytocin released can then attenuate a lot of the anxiety and other aspects that are driving drug taking after the formation of the bond.
Yeah, no, though, that's very compelling, really interesting.
I have another question, if I might, jumping on what Eric said. So definitely there are much more people included female in any kind of experiments these past years. And I read a piece written by Wendy Lynch that has been published a few weeks ago, about the importance of using the right animal model if we want to study female or to compare both male and female. So what is your opinion about that? Do you think it's a shortcut to just include female in our group of experiments and use the exact same model than we use for years for only including males? Should we adapt our animal model, especially in drug addiction?
Well, I think you do need to think about your animal models from the perspective of, is this appropriate for females? And that's why if you think about the social support experiment, where females usually live in a den together with other females, at least most female rodents share housing, while males do tend to be more isolated. So I didn't show these data, but we've shown with cocaine that social housing decreases motivation for cocaine in females, but not males. So as we look at the data, it makes sense. I mean, it's much more stressful, in my opinion, for females to be isolated even female rodents, than it is for male rodents. So thinking about what does it mean that social support works better for females, I think it's what it shows you is that social support is much more important for females because that's what female rodents would have been used to. So I totally agree with Wendy. We need to be thinking about how we measure these behaviors in males and females, because the social conditions, just the perspective is going to be different for the two sexes.
Yeah.
And there's a question in the Q&A now from Carolina Skabika, who asked about natural fluctuations in reward related to the phase of the menstrual cycle or in rodents to the estrous cycle.
Yeah, so we've shown that you do see variation in motivation. If you look at breaking point, for example, in animals in the GPER1 study, the first week they were working on a progressive ratio schedule. We saw a significantly greater motivation during the phases of the cycle, proestrus, estrus, when estradiol had just been elevated compared to diestrus. Mm-hmm. But that was only the first week. And after the first week, methamphetamine wins, or cocaine wins, basically. The hormones aren't modulating the response nearly as much once the behavior is really well established and the amount of drug the animals are taking has escalated. Mm-hmm. So my model is that the hormones of the estrous cycle or menstrual cycle are on board and influence the acquisition and escalation of drug taking. And then once you get to maintenance, the drugs are driving the response to a much greater extent.
Interesting. Yep. Well, that brings us to the end of the hour.
Yeah.
So thank you again so much for covering such fascinating biology and such important work. We really appreciate it. Thank you, Celine, for being co-host today. And please join us again in 2 weeks, as I said, for Dr. Elizabeth Heller from the University of Pennsylvania. Jill, thanks again, and thanks everyone.
Thank you. Yeah, thanks, Celine. Thank you, Eric. Thank you, Eric.